So, so this is the topic of my current research. We are dealing with the phylogenetic patterns of floral scents, and I am particularly interested in the evolution of one very, very awkward kind of pollination. It's a little bit like the dark side of pollination. It is what we call sexual deception, and I will go into detail just a little bit after. A couple of years ago, we've made a review with Jeremy McNeil, another expert in chemical ecology, and we have listed a few spectacular cases where you have some models at the upper part and some mimics. So we have listed cases where, for example, some flowers were mimicking some insects. This is the number one. Some flowers were mimicking some mushrooms. Some flowers were mimicking dung or urine also. Some flowers were mimicking different kind of things. We also found recent evidence for some fungi mimicking carrion, for instance, and also some mosses. So we've been listing all these very interesting cases of natural history. We've been describing the chemistry, and this is another paper I can also send you if you are interested in having a broader perspective on chemical mimicry. So this is the fact where when you have two species interacting with one another, one species is using the chemistry to pretend like it's the other organism. So there are some really, really fascinating cases of chemical mimicry. On the upper left, you see some Asclepiads from South Africa, where you have flies that are attracted by the scent, and the flies, they really think they are getting in contact with an oviposition site, and so they actually lay eggs on the flowers, thinking it's a dead body or it's a corpse or anything like that. One of the most spectacular flowers, it's actually an inflorescence. It's the Amorphophallus that you see on the letter B, and then there are a few more cases of Araceae, among other things, that are also mimicking dead bodies. This you can see on the letter D. As I said, there are also mosses. This is the letter E, mosses that are producing these yellow structures, and they are producing also a scent that is reminiscent of urine. And so they are attracting flies, and these flies, they will be transporting the spores of these mosses. They will be doing like something like the pollination of the mosses, but maybe you know that the mosses have a different biological cycle, so it's a little bit different, but the principle is the same. Some fungi you see on the letter F that are producing this jelly that is very, very smelly, and that's attracting lots of mosses. On the letter G, what you see, the yellow structure is actually a part of a very dark plant that is infected by a specific kind of mushroom. And so this produces a flower-like structure that also emits some really sweet odor compounds, and these attract solitary bees that are thinking they are interacting with a yellow flower. And perhaps one of the most spectacular cases is the orchid that you can see on the letter H. This is an orchid that mimics a structure of a mushroom and also the scent of a mushroom. It's been described more recently in Science. You see, in 2006, the chemistry of these Dracula flowers have been described. And since then, in 2011, we have had some other cases of orchids also mimicking the odor and also the texture of different kind of fungi. So if you have a look at the whole spectrum of weird organisms that are reproducing using chemical mimicry, orchids are very, very special. And within the group of orchids, within the family of orchids, you have all sorts of really, really weird strategies. Some species have evolved very, very awkward ways of attracting their pollinators. So you saw here the fungus gnats that are attracted by the Dracula and also by the other orchid to the left, or to the right, sorry. There are also orchids mimicking what we call green leaf volatiles. These are the compounds that are released by plants that are injured. So when some plants are being eaten by a caterpillar, for instance, these plants will release some really characteristic compounds, and these compounds will attract wasps. So in a way, it's a way for these plants to get protected. So they call for a bodyguard, so to say, when they are being attacked. Well, some colleagues from Germany, they have found that there is one specific orchid that mimics that emits these compounds to attract the wasps. So in a way, it pretends like it's being attacked, but it's not attacked, of course. Here, the orchid just wants to attract the wasp, and the wasp will be collecting the pollen while nectaring, and then they will be transferring the pollen from one flower to the next. Other really weird orchids, again, these ones, they mimic alarm pheromones. So they really hijack the chemical communication systems of insects. And here in these two cases, the first one shows a fly, and it's a fly that's usually predatory on aphids. And so when there are aphids around, sometimes when the aphids on the plants are a little bit stressed, they release an alarm pheromone, and this would attract some flies that will lay their eggs on the plant, and then the larvae of these flies will eat the aphids. Here, the orchid, again, it's pretending like it's a family of aphids, and it's emitting exactly the same compound as the compounds emitted by a colony of aphids. And you have the same thing just below, a paper published in 2009, again, where you have a hornet pollinating an orchid, and the orchid is mimicking the scent of the alarm pheromone of honeybees. But these are the orchids I have been interested in for a bit more than 10 years now. This is a drawing that was made after we published one of our papers, and you can guess what it shows. It shows a male bee that is more attracted by a kind of female, which is actually the orchid flower, instead of its own female. So this was basically the result of a 4-year study that I did during my PhD, and I thought that this cartoon was summarizing pretty well some of my research. So I wanted to show it to you. We know that also in these orchids, we know that the bees, in general, they have something. There's something between the bees and the flowers, you see, and particularly the colors are really important. So most of my research recently has been dealing with the relative role of colors and scent in pollinator attraction. We've started looking at ophris because on some of these orchids, you can see that some structures, here you can almost guess that there are wings and maybe the small antennae on the left. Some of these orchids are a little bit hairy, so they really mimic the body of an insect. And it's not just visual, but it's also with the chemistry, because the scent that they release is also very, very, very similar to the female sex pheromones. And so this is what it looks like. A male is attracted by these orchids. The male tries to mate, to copulate with these orchids. Obviously, it's not very successful, so he's trying to understand how this female works. He's not very successful at mating, but you see the two pollen masses on his abdomen. So from the orchid point of view, this has been really, really successful. The male will fly away with the two sacs of orchid pollen, and then when it will be attracted again by another orchid, it will transfer all the pollen to the other orchid, and this will help, of course, the orchid to reproduce and therefore to survive. So one of the first questions we asked was, is there any particular role of this for this yellow margin on this species here from the Ophris lutea group? But we were intrigued by the fact that the females of these pollinators, they collect the pollen almost only on yellow Asteraceae. So these are solitary bees that are really, really highly specialized. They only collect pollen on the yellow Asteraceae, during their whole life and from one generation to the next. So they are really completely hooked on this group of plant. And the males, they spend most of their time flying from one yellow flower to the next, looking for females. And usually they mate with the females on these yellow flowers. And we know that there's a whole group of really closely related solitary bees, and most of them, they are really, really attracted by these yellow flowers, and most of them, the females collect the pollen on these yellow Asteraceae, and in most of the cases, the males patrol from one yellow Asteraceae to the next, and they also mate with the females on the flower. So again, it's a case of rendezvous flower, as we call it. Here you can see the detail of the 4 different species of Ophrys in this group. You can see that that Ophrys lutea is the big one in the middle, and then you have two small ones on the sides, and there is a 4th one that is not shown here. But most of the pollinators are really highly specific. So you have 2 or maybe 3 species that are regularly attracted by these orchids. And all of these species are really, really closely related solitary bee species. Most of them belong to the same subgenus, so they are really, really closely related. We know that some are the ones that are staying in dark now are polylectic. It means that they also visit a few other plants. The yellow ones are strictly specialized on the Asteraceae, and then there are a few more for which we are still lacking really detailed information. But most of the fieldwork observations relate them to the yellow Asteraceae anyway. So we thought, is there a way of explaining this similarity here between the two? And you can see that the yellow on the margin of these orchids, it really merges, almost it blends perfectly with the yellow of these host plants of the females. And when you compare them, they are really, really, really closely related in the bee color space. So you can see on the figure to the right that we have plotted the color of the yellow Asteraceae and the color of the Ophrys in the same color space. So this is a way to interpret the colors from the bee's perspective, and they are really, really very similar. So now we started looking at the interactions between colors and scent, and we've been looking at one particular interaction here between this solitary bee and the Ophrys species it pollinates. The first thing is that we've been working on one species for which we have 2 color morphs. We have one green morph and a white morph. And so the first question was, is there any floral scent difference between the green morph and the white morph? Is there a difference in the total amount of scent that is produced? Or is there any difference in the relative proportions of the compounds? So the figure to the left shows that there is no significant difference in the total amount of compound that is released. So basically the green morph and the white morph, they release the same amount of scent. And when you look at the proportions, also there is a very high overlap between the 2 morphs. So there is no difference in floral scent between the yellow and the green morph. And so we've been testing this a little bit further in the field. So I have designed this way of testing that in the field. So I used a small piece of plastic on an insect pin. This was my odorless dummy, as you can see on the left. And then I have been adding to this odorless dummy a white odorless perianth, then a green odorless perianth. And then I have used the scent of the orchids on the small pieces of plastic. And then again, I have added to this chemical stimulus, I have also added a visual stimulus to investigate if the presence of a perianth or the color of a perianth would have an effect on the visitation rates. So this is how we do it. We use some extracts of the orchid floral scent, and we put them on these small plastic beads. And then for several minutes, we count the number of approaches and the number of contacts of the male bees with these plastic beads. So the first thing I have tested was just the dummy, and this is just a plastic bead with no visual or no chemical stimulus. Then when we added the white period, well, it didn't really change much. When we added the green period, it didn't change much either. And then when we added just the scent, we observed a dramatic increase in the number of approaches and a dramatic increase in the number of contacts of the male bees. And then when we added a visual stimulation, so either a white period or a green period, there was not much change between all these. So no significant difference when you add a visual stimulus in this case, but the chemical stimulus is really essential. This is really where the high specificity of these plant pollinator interaction lies. So we've done also some electrophysiology. We've done some GCEAD, and this is a technique that we use to ask the pollinator which compound it can smell. So when there is an olfactory receptor on the antenna, and when we blow a compound on the antenna, if there is the right receptor, the receptor will bind with the molecule, and then there will be a small electric current in the antenna, and so we can detect this small current like this between the two electrodes that you see here, and we can record this at the same time as we are recording the different compounds from the extracts. So we get a figure like this. The upper part represents the different compounds we have in the floral scent. So that's the FID. And the lower part, when you have a stimulation, it means that the antenna has reacted to this specific compound. So we looked at the FID traces for the orchids and, of course, for the female bees to see if the female bees and the orchids use the same compounds or maybe different compounds. And what we found is that when you look at all the compounds that trigger a response in the antenna, so we call them the active compounds. When you look at the detail of these compounds, we find that the orchid and the female bees, they use exactly the same set of other compounds to attract the males. So the orchids, they really mimic the female sex pheromone here, and they mimic it relatively accurately because they really release exactly the same compound. They use the same compounds as the female to attract the male bees. So we've been testing that in the field afterwards, and I've been going everywhere in Europe to sample bees and orchids from different populations. And after 4 years of quite intensive field work, we published this paper. I think you have received or you will receive the PDF, where I have been looking at bee populations and orchid populations all over Europe. And I have been collecting the scent and collecting the sex pheromones in sympatry, in allopatry, so in the same populations, in different populations. And when we put all of these together, what we find is that the orchids and the bees, they form two different clusters. So there is very, very little overlap. So it was really strange at first, because we thought if the orchid wants, so to say, to attract these pollinators, then it should work better if the orchid mimics the female bees accurately. But here what we found is a case of imperfect floral mimicry. So we found that there are differences, something that we expected to be completely overlapping, so the same stimulus. Actually, these are two slightly different stimulus. So we've been looking at what happened at the population level, and this is the figure to the right. We've been collecting orchids and bees at the same populations in France and in Italy. And as you can see from the graph, there are differences in the bees from one population to the next. So look at the symbols the open circles and the black circles, you can see that these form two really different clusters. And then the orchids, these are the open squares and the black squares. These also form two different clusters. So this is a case of what we call sex pheromone dialects. So it means that the females use the same compounds, but the proportions of the compounds, they slightly differ from one population to the next or from one country to the next. So we call them dialects. And we found exactly the same thing in the orchids also. And again, we found that the orchids were different from the local bees. And this is the results we got when we tested just the floral scent extracts of the female bees, of the local female bees and the local orchids on the patroning males. We found that the males were more attracted by the local orchids than by the local females. So this is, again, you should remind you the cartoon that I have shown earlier, showing that the males were more attracted by the orchid than by their own females. And so we thought we would look at some of the key compounds for the attraction of the males, 3 compounds, and we looked at the differences here between the model and the mimic, so between the bee and the orchid. We found significant differences in the relative amounts. Excuse me. And when we manipulated, so we changed the chemical composition of the extracts of the female bees to make them smell like a local orchid. And when we did that, it significantly increased the attractiveness of the extract. So it really shows that it's the ratios, so the proportions of the compounds that are really important. And when you change them to make them smell a bit more like the orchids, then the extract become a lot more attractive, significantly more attractive. So the floral scent, clearly, this is the key to pollinator attraction in these orchids. We found that it is a case of chemical mimicry, so there are the same compounds that are used by the female bees to attract, and by the orchid to attract the patrolling males. And we found that the males, they have a strong preference for sex pheromones from other populations. So when you have the males at one population, and when you test the odor of the female from the same population, but also the odor of the female from other populations, well, it turns out that the males are more attracted by the odor of females from other populations. So this is presumably the mechanism behind the evolution of these orchids. So the orchids, they maintain these slight differences in scent, so they don't have to become very similar. So remaining slightly dissimilar increases their attractiveness to the local males. So this is the road to success, of course, and there is a very strong pollinator-mediated selection on these orchids. So they are really selected to fit to the preferences of these females. This was for the office. So I thought, okay, this is really nice. We've been looking at one particular, quite a few species actually, but we've been looking at primarily the genus Ophrys. So look at the phylogeny to the left. You can see that there is one species called Ophrys sphegodes. It's approximately right to the middle. You should see it. So this is just one genus, but in Europe we have several genera like that. And you can see them from to the right. I have been plotting again a phylogeny, and I've also been plotting the different kind of pollination mechanism. And you can see that some orchids, like the gymnadenias and like the platanteras, for instance, they produce a lot of nectar. They are attracting bumblebees, or they are attracting moths during the night. Then you have other species like the Transteinera and the Orchis, or Hemantoglossum neotinea. These are pretending like they are producing nectar, but they are not producing nectar. We call this food deception. So they pretend like they offer nectar to the pollinators, but there is nothing inside the flowers. And then I've been looking at one particular genus. It's called the genus Serapias. You see it? It's just above the Ophrys. And I've been looking at this particular genus because it's really closely related to Ophrys, and I wanted to know a little bit more about what's going on in there, because there was nothing much known about their pollination biology. So let's have a look first at this genus. First of all, I had to remind you that this is from a study from 2008, and they were looking at the odor compounds that we found to be attractive for the male bees. They said, okay, these compounds are really important for Ophrys, but are these compounds specific to Ophrys, or can we also find these compounds elsewhere? And as you can see from the phylogeny, the compounds are found in all of the genera that are being depicted in black. So it's not an innovation of the genus Ophrys. They are really present in trace amounts almost everywhere. So these are the main conclusions of this first study. They said the production of these alkenes is very widespread. So obviously it might be an example of a pre-adaptation. So this pre-adaptation might have helped for the evolution of chemical mimicry. So it's a little bit like you have compounds that are produced by lots of species, and then suddenly there could be selection for an increased amount and then different proportions, and this would be the road to sexual deception. So I have been putting 3 basic assumptions to the test. I have been testing these 3 factors. The first factor I've been testing was the question, is sexual deception something that has evolved from food deception, for instance? Is sexual deception something we find only in orchids, or are there also plants from other families that can do that, especially in our area of the world? And when you reach a stage where the interactions with the pollinators are so specific, so highly specific, this could be very risky, of course. When you rely on only one pollinator, if you lose this pollinator, then it's over, game over. So the question was, is there any way out of this extreme specialization? So we've also been testing that. This would be the third point. Is everything okay for everybody? Everybody understands? I'll just get out of the full screen mode. Yeah? Yes? So the scent, the other compounds, I can show them to you here. So you can have these compounds: alpha pinene, beta pinene, myrcene, phellandrine. These are compounds that are used as alarm pheromones by aphids, but they are also highly attractive for syrphids. And the syrphids, they are also used for biological control, for instance, because the larvae of these flies, they eat the aphids. So here, actually, the fly, it visits the orchid thinking it will find a colony of aphids where it will potentially lay eggs, but it keeps on searching, and then it finds some nectar, and then the pollen masses of the orchids get attached to their body, and this is how, again, the orchids reproduce. Is that okay? Yes? For bees. So for bees, you can buy pheromone compounds, for instance. This is something you can do, and you can test it when you see people with a big beard of bees, for instance. These are people who have put some queen pheromone on their face, and so all the workers, they come to protect what they think would be a queen pheromone. So you can disguise yourself chemically as a queen bee, if you like. Yes. When I'm talking about pre-adaptation, you will see you will see it right now, because I will be dissecting the role of scent and the role of morphology and color in the second part now. Okay. Okay. So these were the three different assumptions that I've been putting to the test. Let's start with the first one. Is sexual deception something that has evolved from food deception in European orchids? I've been putting food deception very clearly here because there are, in many books, you have this common sketch where you have the ancestral state would be an orchid that produced nectar, and then some flowers would have lost the ability to produce nectar, and from these flowers, so from these nectarless flowers, sexual deception would have evolved. This is a very cliche sort of figure from most of the orchid books. This was the road to sexual deception, as it was described in many books and many papers, especially in the 80s. So we know that there are a few cases where there are shifts between food deception and sexual deception, but we are still investigating them. It's still not very clear, but this is one of the cases. Orchis galilea, it's an orchid from the east of the Mediterranean, and it's completely derived from a group of food deceptive species, and we think it is sexually deceptive, but we have no evidence. We don't have strong evidence for that so far, so I'm not going to discuss about all this for now. I'm just going to talk about the Serapias, the genus Serapias. It's a genus of orchids, of terrestrial orchids, and they are very characteristic because they have these reddish, these dark red flowers, and they have tubular flowers. So the flowers, they form a tube, and the inside of a tube is very dark. And usually the flowers are like this. They are hanging a bit, and they are relatively large, and a little bit hairy at the entrance, as you can see. And they are only pollinated, or mostly pollinated, by male bees. And the male bees, they visit the flowers at the end of the day when they are looking for a shelter. So the male bees, they go inside the tubes, and they visit these tubes when they are, at the end of the day, when they are looking for a place to spend the night. So it's a little bit like a hotel for males, if you want. So they are really spending the whole night in these tubes. And this was a shot that I made early in the morning when I pressed one of the tubes. There was a male bee in there. And when the bee, they go to the bottom of the tube, then they get in touch with some sticky masses. And on these sticky masses, there are the pollen masses. So when the male was crawling out of the flower, you can see here that it had a full crown of pollen masses. These are all the pollen. This is all the pollen from several plants that's been attached to the head of this male. So we've been investigating this because it's a very unusual pollination strategy, and we found that it has evolved independently in orchids and irises. And as I said, the flowers are mainly pollinated by male solitary bees. Sometimes we have a few female bees visiting the flowers, but on average there are many males compared to the number of females who can actually pollinate these. So as I said, the flowers, usually they are dark red, they are tubular in shape, and they provide a shelter to the male solitary bees during the night or during the periods of overcast weather. When it's raining a little bit, then they look for a shelter and they visit these flowers. They will never visit the flowers for nectar because there is no nectar and there is no pollen that they can collect and eat. They really only visit these flowers at the end of the day, and during the day there is nobody around these flowers. But there is one exception, and this exception is this species. It's also a Serapias, but as you can see, the inside of the flower is very different. It's very light, and then you have this structure that looks like a tongue, you see? So we call it Serapias lingua, the tongue Serapias. And so I've been investigating this one because I found when I was spending my time in the field that it was visited only by males of only one solitary bee species at the hottest hours of the day. So So this is very different. Most species, they are visited at the end of the day or when it's raining by males that are going slowly and they are getting inside the tubes. These males, they are going completely crazy and they visit these flowers at the hottest hours of the day. They go from one flower to the next. They are completely excited. So I thought that there should be something there, and this is really something we have to investigate to link it to off-risk pollination. So with these flowers, I've been opening the flowers because I found that the males were obviously interested in something from within the flowers. So when I artificially opened the flowers, I could see that the males, they were obviously very interested in this tongue-like structure. Sometimes they were queuing one after the other, so that they were coming just waiting one after the other to see what's going on at the bottom. And then I thought, okay, so if this is really the thing they are interested in, then I'm going to rip off the whole flower and just keep this, you see? So I've really been destroying the whole flower, and then I've been presenting this to the males, and believe it or not, they were completely crazy about it. So they really, they were turning around and upside down. And so I've been also filming them. And although this is a very slow motion version of a copulation attempt, the males are really trying to mate, to copulate with this structure. And so it's really the tongue-like structure of these orchids that is really important here. So you see it's a little bit less energetic than the off-risk pollination sequence, but nevertheless, this is bee sex. So then I was thinking about my previous studies on the scents and on the way that I had been deconstructing the morphology and the scent. So again, I've been testing just the control, so just the plastic bead, then an intact flower of Serapis lingua, then a flower that was completely ripped off with just the tongue, and then the floral scent extract on a plastic bead, and then also a body extract, so the odor of the body of the female on the plastic bead. And you can see there is no significant difference between these four stimuli, and these four stimuli are significantly more attractive than the control. So obviously there is something about the scent of these orchids that makes it very, very, very similar to the body odor of the female extract. So look at the figure. You have the floral scent extract and you have the female extract. These are the two chemical stimulus, one from the orchid, one from the female, and there is no significant difference in the attractiveness between these two extracts. The color is also very, very weird because it's dark red. So I thought, can bees see dark red? Is it something meaningful for bees? So I started making some reflectance measurements. So I used a spectrophotometer, and then I've been measuring the reflectance of the abdomen of the female and also of the tongue-like structure, because obviously the tongue-like structure looks very much like the abdomen of the female. They are very, very similar. You can see the gray line and the red line. They are almost completely overlapping until 630 nanometers, where you have Serapis lingua going straight up, and you have Ceratina cucubitina going approximately the same way. This is really interesting because the point where these two stimulus diverge, this is exactly the region where the bees cannot discriminate between stimuli. So from 630 nanometers to 700, it's all the same thing. It's all black for the bees. You see? So dark red is a color that is perceived as black from the bees' perspective. So the bees, they don't see a difference between black and dark red, provided that there is no reflectance in the ultraviolet between 300 and 400 nanometers. So here we have these two signals again overlapping in the bee color space, showing that they are very similar. Let's have a look. Let's zoom out a little bit from Serapis lingua. Let's look at the whole group of Serapis again. from the morphological perspective, we have three different groups. We have one group where there is not a tongue, but just two subparallel ridges like this. They form a kind of a gutter inside the flower. So remember, this is really at the bottom of the tube. So these form just a gutter. Then there is another group where the two ridges, they fuse towards the apex, but they still form a gutter. And then there are two species where the gutter is completely fused and they form a tongue-like structure. And the perfect tongue-like structure is only found in Serapis lingua. And the other species where we have a similar structure, it's a self-pollinating species. So this has nothing to do with pollinator attraction, apparently, in this species. So you see Serapis lingua, also from a morphological perspective, it's also coming from a stock of species where we had two ridges that have fused progressively until they form a tongue-like structure. And this is the structure that the male solitary bees are really interested in. Let's have a look at this tongue-like structure again. The width between the abdomen and the orchid structure. The orchid structure is just a little bit wider, and the orchid structure is just a bit longer. So it looks like the tongue-like structure looks like an abdomen of a very fit female from the bee's perspective. And also, if you have a look at the tube of the flower, it's also slightly larger than the nest entrance of these bees. Why do I mention the nest entrance here? How does it fit into the picture? Where if you look at these bees early in spring, what happens is that these bees, they are nesting in hollow stems. So they are looking for stems with a hollow in them. And this is where the females, they make their nest. And the males, they are patrolling from one hollow stem to the next. And when there is, in these light stems, when there is a black patch in there, then they visit the stems, and sometimes they take the female inside the nest and they take the females out of the nest to mate with them. So the males, they are naturally attracted by a tubular structure with a light interior, with a white interior, and with a tongue-like or with an abdomen-like structure in there. This is one of the stimulus they use to detect the females. They also use the scent, of course, but from a visual perspective, this is what they are looking for. If you have a look at the position, the phylogenetic position of Serapis lingua inside the genus Serapis, this really confirms what I've just said before. The fusion of the two ridges into a tongue-like structure, it really happened this way. So we really have the ancestral state would be two subparallel ridges that fuse together to form a tongue-like structure that is only found in Serapis lingua. And Serapis lingua is in a group of tetraploid species, so we still have to figure out what the polyploidy, what the role of the polyploidy would be in this system. But what's really important here is that Serapis lingua is derived from a whole stock of other species, and all the other species, they are pollinated by shelter mimicry. So they are mimicking a shelter for the male bees, but Serapis lingua is pollinated by sexual deception. So you see the picture here. We have an evolution from shelter mimicry to sexual deception. This is what I have illustrated here. In the genus Serapis, we have the evolution from shelter mimicry to sexual deception, and sexual deception is a derived condition, so something that has appeared more recently in the course of evolution. If you have a look at, again, the key compounds for male attraction, if you have a look at the alkenes, these are really the important compounds. If you have a look at the alkenes in white versus the alkanes in black, the alkanes are not so important. They are just physiological compounds to prevent the water loss of the flowers and of the leaves. But the alkenes, so the white ones, are very important because these are the key compounds for mate attraction. Look at the proportions of these two classes of compounds. When you go from a species like Serapis cordigera, which is pollinated by shelter mimicry, when you go from that to species like Serapis lingua pollinated by sexual deception, you have a significant increase in the alkenes, so an increase in those compounds that are responsible for the attraction of the male bees. Second question. We hear and we read a lot about sexual deception all around the world now, because we have instances from South America, also from South Africa, from Australia, also from Europe. We thought we would be asking the question of whether sexual deception is only found in orchids in Europe, or if there are other plants in Europe that are also pollinated by sexual deception. So again, I've been looking at other plants that are also pollinated by shelter mimicry, and these are the royal irises from the east of the Mediterranean basin. These are really superb flowers, and they all have this black disc just at the entrance of the tube. You see, this one is very colorful. Some of these species are almost black, and they flower in the middle of the desert very early in spring. And this is one of the species I've been working on also. Again, you see it's dark red, and you remember what I said. The dark red is really perceived as black by the bees. So this is how it happens early in the evening or very early in the morning. What you see is that there are male solitary bees sleeping one next to the other. You can see all the pollen grains on their body. And so they are transporting these pollen grains from one flower to the next when they are looking for the right shelter to spend the night. We know that the color, this dark flower is very important. We know that the males, they look for these shelters because some of the color mutants of these irises, they have absolutely no reproductive success. So color seems to be very important, and a dark shelter is something they really look for at the end of the day. For the chemistry, remember I told you about the alkanes with an A and alkenes with an E. The alkenes are the sex pheromone mimicking compounds. Here the alkenes are really kept to the minimum, and the main compounds are just basic alkanes. So there is not much about sexual deception in here, and indeed we found that it's pollinated primarily by shelter mimicry. And we also found that the floral scent of this iris was very similar to, it actually shared many compounds with the floral scent of some of the Serapias species. And we also found that the role of the scent here was presumably not very important. Look at the figure to the left. This shows the floral scent differences between flowers that are opening towards different directions. So there are flowers facing the north, flowers facing the east, facing the west, facing the south, and then all the intermediates. There is no evidence for a particular floral scent linked to one particular flower orientation, and apparently the flowers that are visited, they don't have anything special in their floral scent. As you can see on the right part of the figure, the visited and the not visited flowers completely overlap. So there is no significant difference between the flowers that are visited and those that are not visited from the chemical perspective. So the floral scents here don't seem to be very important, but the colors seem to be very important because the dark flowers are the ones that will be attracting the male bees. But I've been looking at one particular iris in this group, and this was the very first iris that I've seen in this group of night sheltering, of shelter mimicking species. There is one species, and when you look at the identification keys, it's very funny because they say, oh, this is the first species that keys out because it's so different. And so they say, if you look at this citation from Brian Matthew, he says, it's immediately recognizable in any of its form. The flower is remarkable in that the nearly horizontal folds are very small. Indeed, when you compare this one with the dark red that I've just shown you before, they are very different. And so you can see it here. So the regular shelter mimicking species would be the one on the left, and the weird one is the one to the right. Okay? I've been really attracted by the one to the right because the look of the flower reminded me so much of some of the off-root species that I know from the Mediterranean. Look, this is to the right. This is an off-root species that is only pollinated by sexual deception. And to the left, this is the iris for which there was nothing known about its pollination biology or floral scent just a couple of years ago. So I've been making lots of contacts, and I've been in the field also with several guys, and then we've ended up filming the pollination process of this iris to the left, and this is what it looks like. It's a giant carpenter bee, a Xylocopa, and it's only males, and the males are attempting copulation. They are really trying to copulate with these floral parts of these irises. I'll just put it again so you can see the whole sequence again. So they are not looking for nectar. They are not looking for pollen. Although they transport pollen, you can see the white pollen on its thorax. But all they are interested in is to try to mate with these flowers. Interestingly, this giant carpenter bee you've just seen is also the pollinator of one very narrow endemic species from one island in the Mediterranean. It's a huge ophrys flower, one of the biggest we know, and it's only pollinated by these giant carpenter bee. So I've been there in Crete, so in this island of the Mediterranean area, to investigate and to compare the scent of these two. If these two species are pollinated both by sexual deception and both by the same species of giant carpenter bee, then there must be something in their floral scent. And indeed, this is a project we are still running at the moment. We are comparing the scent of these irises with the scent of the orchid and the scent of the females, and they share many compounds, although the proportions of the compounds are different. But you can see from the graph, all the compounds that are numbered are the ones that are shared by at least two of the three species. So this is something we are still investigating at the moment, and we think, again, we have the independent evolution of sexual deception towards the same wild bee species and using chemical mimicry from two different families. Again, if we have the same approach, look to the right, you can see the phylogeny of these irises, and I have been plotting the pollination strategies. We can see that it's the same situation as in the genus Serapias. The sexually deceptive species has evolved from a stock of shelter mimicking species. So the ancestral state in this group of irises is a plant that was pollinated by shelter mimicry. So again, you have a transition between shelter mimicry and sexual deception. It's the same story as in the genus Serapias. So there are many parallels between these two groups. But the story is not over. I've been looking at, again, of course, the alkanes versus the alkenes. And you can see that in Iris paradoxa, look at the white bars. There is a significant increase in the alkenes. This is linked to the evolution of sexual deception, so the need to have more alkenes for mate attraction. And you can see also that in the scent of Iris atropurpurea, there is almost no alkenes in the floral scent, almost just the alkanes, the black bars. So the third question we asked was, is there any way out when you get so highly specialized, when you only attract only the males of one particular wild bee species? Is there any way out in case something happens? And so I've been looking at another group of a really awkward off-risk species. We know that the off-risk species are often mentioned in textbooks because they are one of the most highly specialized plant pollinator interactions. So we know they are very highly specialized, and we know that there is at least one way out of this high specialization, and this way out would be self-pollination. The bee orchid, it's called the bee orchid, but it's a self-pollinating orchid, and it has these elongated pedicels here that you can see. Even Darwin described this bee orchid, saying that it is impossible to doubt that these points of structure and function, which occur in no other British orchids, are especially adapted for self-fertilization. So we call it the bee orchid, and it's a shame because we see the bee orchid in all the books talking about the or writing about the high specificity of off-risk pollination, and they show and they mention the only species in the genus that is self-pollinating. So anyway, that's another story. It's something we have to deal with from time to time. So this would be one way out of extreme pollinator specialization. But then I've been looking at one particular group of closely related species from Greece, so again from the Mediterranean area in Europe. 5 species, and there is one species that pops out immediately. It's the one to the bottom to the right, Ophrys helenae. So it's Helen's orchid, and it's the only one in the group that lacks this H shape in the middle. So the mirror is not present on the flowers, and it's a species that is not very well known because it's strictly endemic to the north of Greece and to Albania. It's not exactly the kind of area where people go on holidays, as some of you might imagine. So it's not a very popular area. I have family in Greece, and when I told them that I wanted to go close to the Albanian border, they asked me, "Are you sure you really want to go there?" But anyway, I went there to spend a few weeks to investigate this orchid, and I remember I had this small paper of less than 1 page in German, published in a very obscure journal, saying that maybe this orchid would also be pollinated by shelter mimicry, but there was very weak evidence. So I thought, let's go back to the field and let's see if we can find the same thing that had been found something like 20 years ago. And I've been back there 2 years ago, and again I found the same pattern, the same functional group of pollinators visiting the flowers only late in the afternoon, and only to sleep on the flowers. So no copulation attempt, absolutely no copulation attempt. They just crawl one on top of the other, and they sleep like this on the flower. So I thought, here we have it. This is an off-risk species pollinated by shelter mimicry and not by sexual deception. Again, I've been making some reflectance measurements, and again you can see. Look at the figure to the right. You have 3 curves. You have one purple curve, a blue curve, and a green curve. These are the 3 visual receptors of the eyes of bees. You see, so they have high sensitivity in the UV. This is the purple curve. High sensitivity in the blue and in the green. And in the area to the right of the dashed line, so from 630 nanometers to 700, you can see that we have an increase in the reflectance. This is just a signal that we have something red. But this is exactly the area where even the green receptor is very, very weak and not very sensitive. So again, this shows that these red flowers that we perceive as red, from a bee perspective, they are perceived as a black hole. We've been describing that in a paper on these irises, because the parallel was again very obvious. So everything I've just said now is also in one of the papers we published last year. This is what it looks like. So we thought, okay, we have one species that is pollinated by shelter mimicry, but is it a derived species, or is it a more basal species? Is it a recent evolution of shelter mimicry from a stock of sexually deceptive species, or is it the other way around? So we needed again a proper phylogeny to do that, and not just the phylogenies that have been published 15 years ago, but at least on some of those we had some hints. So we started like this, and we made this more recent phylogeny. I'll show you the phylogeny just later. But then, of course, the aim was also to compare Ophrys helenae with its sister species to see how different they are. So we've been extracting the scent, and this is the floral scent variation between the shelter mimicking species in orange and the sexually deceptive species in blue, you can see that there is not much variation. I mean, the shelter mimicking species is not significantly more variable or significantly less variable than the others. It's in the same variation on a theme, so to say. And when you look at the real compounds in there, well, it looks like Ophrys helene. So this is the dashed squares to the bottom of the figure. This is Ophrys helene, the shelter mimicking species. All the other ones are the sexually deceptive species. And you can see that it's just slightly different, but it's not like two worlds apart or so. So they are just slightly different. Again, you can see here the shelter mimicking species compared to all the sexually deceptive species is just slightly different. There is not a big chemical gap, so to say. They are more or less the same. So the changes in the floral scent composition between Ophrys helene and the other species is very, very weak, which shows that it's very important not just to have the right chemistry, but also to have the right morphology and the right structure, because you could have something like the right scent, but if you don't have the other right stimulus, then it's not going to work. And this is exactly what happens here. You could imagine that you have more or less the right scent to be a sexually deceptive species, but the structure and the color of the flower is so different that the flowers are not approached as they would be if they had a mirror, if they had more hairs, if they were a bit more complicated morphologically like the other ones. So the macula and the morphological structure of the labellum are really very important in this case. Look again at the complexity of the alkanes and the alkenes. You can see that only Ophrys gregoriana, the top one, this is the one pollinated by the giant carpenter bee. It's a little bit more complex. You can see that there are a lot more compounds that are present. But the other ones, if you compare Ferromequinum, Mimosa, Hans Renardi, and Helene, they have approximately the same profile, approximately. So presence, absence, it's about the same signature. So there was one more step, because now we know that Ophrys helene, look to the right of the screen, Ophrys helene, it's pollinated by 2 or 3 species of the males of 3 species of bees that are sleeping on those flowers. The thing is that in the same areas in Greece, the same pollinators are pollinating other Ophrys species during the day by sexual deception. So in Greece, you have Ophrys helene growing together with Ophrys bombiliflora growing together with Ophrys eldrachii, and Ophrys bombiliflora and eldrachii are visited during the day. They are pollinated by sexual deception. And in the same meadows, you have Ophrys helene pollinated by the same males, but through a system of shelter mimicry. So again, I thought, look, this would be very interesting to compare, because we have no idea if the scents would be, again, very similar or very different. So again, I've been comparing the traces, and this really shows that Helene has kept the floral scent chemistry of a sexually deceptive species. You can see here there is again almost the same signature between the 3 species for the presence and absence, but again the morphology is very different and the color is also very different, and this is really what makes it particularly special and particularly perceived as a flower mimicking a female bee. Again, if we look at the compounds, look at the differences. So the triangles are the shelter mimicking species, and the open discs and the black discs are the sympatric sexually deceptive species. These 3 could be sister species. I mean, they are very, very similar chemically speaking. And when we look at the phylogeny, here we go with the phylogeny. The story is very different from the first 2 cases, because here we see that Ophrys helene is derived from a stock of sexually deceptive species. So the root went the opposite direction, and here there is a way out of extreme specialization, and this way out would be the pollination by shelter mimicry as is shown in Ophryselidae. And again, as I've shown here, there is no much difference between the alkenes and the alkanes when you compare the sexually deceptive species and the shelter mimicking species. So this is again in contrast with the first two cases that we have seen. So you see that all of these species, they belong to different genera, even to different families, but they attract the same functional guilds of pollinators, and presumably these pollinators, they exert relatively strong selection pressures on the floral morphology and also on the floral sense, and this drives phenomena of convergence and divergence that are quite spectacular. In the same region, you have species of different families that evolve towards the use of the same pollination strategy and even the same pollinator as we've seen for the giant carpenter bees. And you see also purely from the reflectance point of view, we have really the same kind of traces. So all these dark red flowers are really perceived as completely black by the pollinators. And you have more or less this situation when you have a transition from shelter mimicry to sexual deception, you have a significant change in the proportion of alkanes versus alkenes. The alkenes rise because these are the compounds that are mimicking the sex pheromones. So this is the summary. So I told you I had three basic assumptions that I wanted to test. Is sexual deception a strategy that has evolved from food deception? Well, not really. We have more convincing evidence that sexual deception has evolved from shelter mimicry in orchids. Is sexual deception something we find only in orchids in the West Palearctic region? Not really. We also found the irises were engaged in the same sort of obscure pollination strategies. And there is a way out of extreme specialization, and this would be shelter mimicry as shown by the pollination biology of Ophryselidae. Just a few acknowledgements of the people, because this represents a few years of field work a bit everywhere, lots of interactions with lots of botanists, with lots of amateur botanists who are extremely knowledgeable. A few students who also helped me in the field, and a few funding partners, because if you don't have any euro cent, there's no way you can travel around. So I have to acknowledge them, and I thank you for your attention. I will just exit the full screen mode so I can see you if you sleep or if you are ready for a few questions. Thank you.